Non-Mendelian inheritance. Not always a simple relationship. The relationship between genotype and phenotype is rarely as simple as in the pea plant characteristics Mendel studied. Many heritable characteristics are not determined by only one gene with two alleles. However, the basic principles of segregation and independent assortment still apply even to more complex patterns of inheritance. Deviations from Mendelian genetics. Inheritance of characters for a single gene may deviate from simple Mendelian patterns when alleles are not completely dominant or recessive, a gene has more than two alleles, or when a gene produces multiple phenotypes. Complete versus incomplete dominance. What we have seen until now when studying Mendel's work is complete dominance. This occurs when the phenotypes of the heterozygote, for instance big P little p, and the dominant homozygote, big P, are identical. This is not the case in incomplete dominance. In incomplete dominance, the phenotype of the F1 hybrids is somewhere between the phenotypes of the two parental varieties. In this case, we have white flowers crossing with red flowers to produce pink flowers. Blending versus incomplete dominance. This third phenotype results from the heterozygote having less red pigment than the red homozygote. This is not blending. If blending occurred, you would never be able to achieve the red and white traits from the pink hybrids. This is not the case. It is possible to obtain red or white offspring after crossing the two pink hybrids. So we want to stay away from this word blending because we don't want to imply that the actual genes get blended. Even though the colors look like they're mixing together, we would rather say that it's just having less red or less white. It's pink, it's somewhere in the middle, but we don't want to say blend. Codominance. In codominance, two dominant alleles affect the phenotype in separate distinguishable ways. One of the best examples of this is the ABO blood group phenotypes in humans. This involves a single gene with three different alleles. These three alleles, in different combinations, produce four phenotypes, which are the common blood types that we know about: type A blood, type B blood, type AB, and type O blood. The relationship between dominant and recessive alleles. So what is the relationship between dominant and recessive alleles? A dominant allele does not subdue a recessive allele. Alleles don't interact. Alleles are simply variations in a gene's nucleotide sequence. For any characteristic, the dominance or recessiveness in relationships of alleles depends on the level at which we examine the phenotype. Click here to learn why dominant alleles are expressed over recessive alleles. Pleiotropy. Pleiotropy is a situation in which a single gene may affect many phenotypic characteristics. Pleio means more than one, and tropy means affecting or pertaining to. An example of pleiotropy in humans is sickle cell disease. Sickle cell disease. This is a disease characterized by many diverse symptoms such as abnormal red blood cells, leading to physical weakness and anemia. So if these blood cells are abnormal, they can't carry as much oxygen, causing that physical weakness. Clumping of cells, and clogging of small blood vessels leading to pain and fever, possible brain damage, and possible heart failure. So if you look at the shapes of these cells that are crescent shaped, they're not as round and able to flow through the blood vessels as well, so they clump more and then they stop blood flow, leading to these symptoms such as possible brain damage and heart failure. Accumulation in the spleen of abnormally shaped red blood cells leading to spleen damage. The spleen normally removes red blood cells that have lived their life and are worn out, and so if these crescent-shaped blood cells get clogged up in there, that could be a big problem for the spleen. Sickle cell disease. All of the possible phenotypic effects of sickle cell disease are caused by the presence of a single allele when it is present on both homologous chromosomes. And this single allele, by the way, is a mutation that causes just one amino acid to be wrong in the whole amino acid sequence of the protein. The direct effect of a sickle cell allele is to make red blood cells produce abnormal hemoglobin molecules, which tend to link together and crystallize, again due to that one wrong amino acid being in that hemoglobin. This in turn leads to abnormally shaped red blood cells, which take on that crescent shape. Polygenic inheritance. Another variation from Mendelian genetics is that a single characteristic may be influenced by more than one gene. Mendel studied characteristics that were classified as either or, such as purple or white flowers. Mendel's pea plants were either tall or short, there was no in between. Each characteristic depended on only one gene. It does not always work that way in genetics. Many characteristics vary over a large scale in a population and are determined by many genes. Poly means many, and genic refers to genes. Human height. What governs height in humans is actually the additive effects of two or more genes on a single phenotypic characteristic. Human height is governed by multiple genes. Polygenic inheritance is the opposite of pleiotropy, in which a single gene affects several characteristics. Modeling human skin color. Skin color in humans is controlled by at least 3 genes that are inherited separately. For simplicity, we will stick with 3 genes in our example, and these 3 genes are big A, big B, and big C. Each of these genes has 2 alleles, a dark skin allele and another allele. The dark skin allele for each gene, big A, big B, or big C, contributes 1 unit of darkness to the phenotype and is completely dominant to the other allele, little a, little b, or little c. Modeling human skin color. A person who is genotype big A, big A, big B, big B, big C would be very dark, while a person with genotype little a, little b, little c would be very light. A heterozygote, which is big A, big B, little b, big C, would have a shade of skin that is intermediate to the first 2. Since these alleles have an additive effect on the phenotype, a person who is heterozygous, big A, big B, big C, would have the same skin color as any other genotype that has only 3 dark skin alleles. For example, big A, big B, and little c. Modeling human skin color. As calculated by our simple model, the proportion of the population according to skin pigmentation. The proportion of the population is on the y-axis, and the skin pigmentation is on the x-axis. Notice, most people have a shade of skin color that is intermediate to the extreme skin colors of their parents. Modeling human skin color. Our simple model illustrated how 3 different genes could produce 7 different skin color phenotypes in humans. However, humans have many more skin color shades than shown in the model. A more precise model would give a better representation, but not the complete story. Environmental factors such as exposure to the sun, geographical location, and diet strongly influence this trait. Heredity and environment. Many characteristics result from a combination of heredity and environment. A tree's phenotype is genetically determined, but its environment will influence the phenotype as well. Exposure to wind and sun will affect leaf size, shape, and color.